So what genetic evidence do researchers have they can bring to bear on these two models to determine which is correct? To answer that question, I need to go into a little detail onto a very interesting evolutionary story that you may or may not have heard before. There are organelles inside your cells called mitochondria. And believe it or not, they used to be free-living bacteria. The story goes a little bit like this. A very long time ago, there was a prokaryotic cell, a cell without a nucleus, that evolved these infoldings of the plasma membrane that then became internal organelles, and particularly a nuclear membrane. And that was the origin of the first eukaryotic cell. But then some very odd things happened. I mean, this cell formed its own internal organelles, but it also started picking up some passengers. One of the passengers that got engulfed by this early eukaryotic cell was a prokaryotic cell, a cell without a nucleus, that was an aerobic heterotroph. Aerobic means that it's an organism that needs and uses oxygen, and a heterotroph means it's an organism that can't make its own food. So what you have is some sort of organism, some free-floating bacterium that has to acquire its food from outside of itself. It can't make its own energy. But when it acquires high-energy chemical compounds, it breaks them down using oxygen. And this is a very efficient way of metabolizing high-energy chemical compounds, because when you use oxygen as an electron acceptor, you can get a lot more energy out of those compounds. So what ended up happening is that this early eukaryotic cell engulfed one of these prokaryotes, but instead of eating it, it basically just made a home for it. And this early prokaryote became what's called an endosymbiont. That's a organism that lives inside another organism, but not as a parasite. The two are basically getting along, involved in a cooperative venture together. And this cooperative venture turned out to be very, very successful. The larger eukaryotic cell provided food to the smaller prokaryote. The prokaryote then broke those food molecules down very efficiently using oxygen and gave some of the resulting ATP, some of the resulting energy, back to the larger host cell. Over time, this association between the eukaryote and its smaller passenger became so close that neither one could live without the other. In fact, a lot of the genes from this prokaryote ended up getting moved into the nucleus of the larger eukaryote. Although even to this day, the small prokaryote has its own little tiny circular genome, much like the genome of any free-living bacterium. We call these little passengers here mitochondria. They're an integral part of our cells. So every cell in your body actually has two separate bacterial ancestors. One, the original eukaryotic cell, and second, the mitochondria that became engulfed by it somewhere along the way. Chloroplasts in plant cells, by the way, evolved by a very similar route. They used to be free-living autotrophic prokaryotes that were engulfed and entered an endosymbiotic relationship with a eukaryote that already had mitochondria as passengers. The reason I bring all of this up is to mention mostly the fact that mitochondria have their own genome. In addition to all the genes that you carry within your nucleus, there's a smaller genome that is found within each mitochondrion in your body. Mitochondria don't mate. Each new mitochondrion is an exact genetic clone of its parent mitochondrion, minus any mutations. And you don't receive the mitochondria in your cells from both parents. The mitochondria in your entire body are the descendants of the mitochondria that you inherited in the egg cell that was made by your mother. So when that egg cell was generated, it contained a few of your mother's mitochondria, and they were passed on And all of you, or the mitochondria in your whole body, are essentially an exact genetic copy of your mother's mitochondria. If you are a male, you will not pass on any of your mitochondria to your offspring. Mitochondria are only passed on from mother to daughter to daughter to daughter, and so on, down through the generations. And so when you look at mitochondrial DNA, what you're doing is you're looking back maternally, and only maternally, back through many, many generations. Mitochondrial DNA, or as it's often referred to, mtDNA, is frequently used in genetic studies. And here's one of the ones that caused kind of a stir when it came out. What these researchers announced, they had this catchy name for it. They announced that they had found mitochondrial Eve, the woman whose mitochondria ultimately gave rise to all the mitochondria in all human beings in the entire world. Basically, what this is, it's the most recent common ancestor for all living mitochondria. Or another way of putting it is that if they went back far enough and they estimated that this is the date when the coalescent of all mitochondrial genomes in the human species can be found. Now, I do want to point out that this was not the only human female living around that time, or the only set of mitochondria living at that time. It just is the one mitochondria that ultimately gave rise to all of the different mitochondria in all human beings today. When they did this coalescent, you can see the graph here, their estimate is that mitochondrial Eve lived about 170,000 years ago. You can use similar techniques on all sorts of different genes to estimate the coalescent for any locus in the human species. And what it looks like is that the most recent common ancestor of all modern humans at all loci, or almost all loci anyway, is at about, dates back to about 200,000 years ago. So using the understanding of the coalescent and using the molecular clock, these researchers estimated that all of the genes that we see today, with a few interesting exceptions, which can be traced back to human beings that lived about 200,000 years ago. Now, this is what you would expect if the African replacement model were correct. If the multi-regional evolution model were correct, you'd expect a much older, most recent common ancestor. And you can see that on this tree here. According to this diagram, the most recent common ancestor of all of these tips is right here. It's fairly recent. According to this diagram, if you trace back all of the alleles that are found here at this present day, you're going to have to go all the way back here to find the common ancestor. Okay, and this is about 1.5 or 1.8 million years ago. This is only a couple 100,000 years. Okay, another piece of information that supports the African replacement model is that you find more genetic diversity between African populations. So that again is consistent with this model. These two populations, and maybe they are, the Dinka and the Nuer, split apart from one another a very long time ago. And so when you compare them in the present day, you'll see more genetic differences between those two populations than you would if you compare, say, any other two populations of people that are outside of Africa, like, say, the Scandinavians and the Germans. And the reason you see more difference between the Dinka and the Nuer than you do between populations outside of Africa is the molecular clock. They split apart a longer time ago, and so they've had longer to diverge. And these differences still show up despite the fact that you do have a lot of gene flow among these different branches. Okay, with a multi-regional evolution model, you don't expect to see a big difference when you look at individuals genetically within one region or another of the planet, because all of these branches go back a long, long way. And according to this model, there was a lot of gene flow mixing things up, so that genes that arose in Africa and genes that arose in other places from Africa, according to this model, they all got mixed around pretty much randomly across the world. Okay, however, something that's interesting is that we do find genes in modern human beings today that are descended from genes that evolved first in, for example, the Neanderthals, or Homo heidelbergensis, or there's another group of very mysterious early humans called the Denisovians. And some of their genes survive to the present day. The African replacement model doesn't predict that, but the multi-regional evolution model does. And this is one of these cases where really these are two extremes, and the truth is probably somewhere in the middle. Probably the history of our species is something more like this. This is called the primary African origins model. And what it really is the African replacement model with a little dash of multi-regional evolution thrown in. Basically, according to this model, modern evolutionary or anatomically modern human beings evolved about 200,000 years ago. And then they spread out, pretty much replacing everything else. But there was some interbreeding along the way. And so some genes from, say, Neanderthals made their way into the gene pool that survives today. Okay, what else can the genetics of human populations today tell us about the history of our species? Quite a lot, actually. This is an interesting graph, and it's looking at the genetic diversity in different populations as a function of how far are they from East Africa. And the different colors are different regions of the world. The populations from Africa are shown up here in black. The populations of the Middle East are the blue. They're very close in here. Lighter blue is in Europe. Asian populations are here in green. Then those in Oceania, that's places like Polynesia, the Pacific Islands. And finally, populations that are native to the Americas, indigenous peoples in the Americas. And this is the genetic diversity. It's a measure of essentially how genetically different two individuals within any one of these populations tend to be on average. The upshot here is that if you sample a population of people, say the people living in a small town in Africa, you'll find that there's a lot of genetic difference from one individual to next. If you do the same thing, say, in Peru, if you look at an indigenous population in Peru, you'll find that individuals there are much more likely to have the same alleles. Okay, one of the mechanisms that might explain this is founder effects. Okay, and there are two ways that founder effects might produce this sort of pattern. Let's take a look at these two hypotheses. One hypothesis is that the genetic diversity you see in each of these regions today reflects how many individuals first colonized that area. And you should remember that when there's a founder effect, the smaller the group that migrates and colonizes a new area, the fewer alleles they're going to carry with them, and subsequently the lower the genetic diversity of that population is going to be. So remember that a founder effect is just a very extreme example of genetic drift, and that small populations lose genetic diversity very rapidly. Okay, according to this hypothesis, there are 4 groups of modern humans that each spread out independently from Africa. One went straight to Asia, and that population that went to Asia was slightly smaller than the group that went from Africa to Europe. And so there's less genetic diversity that got carried to Asia. It happened to be that the groups that went and colonized Oceania and the Americas were smaller still. And so when you look at all those populations together, what you're seeing is basically how big was the group that founded the population of that part of the world. This was the biggest, second biggest, and then smaller ones out here. Okay, that would explain the data that we see here, this decrease in genetic diversity as you move away from Africa. A second hypothesis is you have what's called a serial founder effect, that you have a group of colonists go from, say, Africa to Europe, and then a group of those colonists migrates to Asia. Some of the group that migrated to Asia, some of their descendants spread out to Oceania, and some of the descendants that find themselves in Asia end up spreading out to the Americas. Basically, what's happening here is that every time a new set of colonists set out, they're only bringing a subset of the genetic diversity in a particular population with them. And so the reason you have less genetic diversity in, say, Europe than you have in Africa is because the colonists that went to Europe weren't carrying all of the original alleles in Africa. When individuals went out from, say, Africa to colonize Asia, according to this hypothesis, they only brought a subset of the European alleles with them. Then when individuals went off to colonize the Americas, they only brought a subset of the Asian alleles with them, and so on. So that you get this, it's a sequence of founder effects, one after another, that have led to the pattern that we see in the present day. Okay, these are the sort of things that people who think about migration and genetics think about. How do you decide between these two hypotheses? Well, remember that the principle of parsimony suggests that the simplest explanation should be preferred. If you think about this in the terms of homoplasies in phylogenetic reconstruction, you should remember that what we really want is the explanation that requires the smallest number of coincidences. So, which one of these hypotheses requires fewer coincidences? If you said it was the serial founder effect, that's correct. You'll notice that, and I phrase it this way on purpose, you'll notice that this hypothesis up here has a lot of happened tos. Okay, in order for these groups to end up in this sort of smooth declining line as you move away from East Africa, the sizes of the groups have to be in just the right order. Okay, and that's not as likely as the fact as that you might have just had a serial founder effect. If this is what happens, then the decrease in diversity just happens step by step automatically. It doesn't require the groups to be in some particular order in terms of size. And so this is likely why we see the trends in genetic diversity that we do. And in fact, when we couple this with archaeological data, it turns out that this is pretty much tells the story of how the globe was colonized by human beings. You have movements out of East Africa into the Middle East, and from there into Europe and Asia. Then from there you get individuals moving out into Oceania and later on into the Americas. And at each step, you lose some of the genetic diversity that was at the previous location. And so you get this decline in genetic diversity as you move away from where modern human beings originated. You can use some of the data we've already seen to figure out when all of this happened. The coalescent for all human DNA is back here at about 170,000 years ago. But this in here is the clade that from which arose all of the people outside of Africa. So if you look at people who are anywhere on this planet other than Africa, they're all going to cluster in this part of this very, very dense tree. And their most recent common ancestor is around maybe 60 or 65,000 years ago, implying that that's when the migrations out of Africa that took over the world started to occur. The nifty thing is that we've got more genes than just mitochondrial genes in our genome. After a while, another study looked at human Y chromosomes, which are only inherited from father to son, and figured out when mitochondrial Adam existed. Similarly, the study here suggests that all non-African human Y chromosomes date back to about 40 to 60,000 years ago. And if you look at genes for markers all across the human genome, thousands of them, you get an average date for the coalescent of about 55,000 years ago. This is another example of how incredibly powerful genomic technology has become. It took years and years for researchers to put together that study on mitochondrial Eve, and that's just for one gene. Now it's very simple for anybody with a fast computer to take publicly available data and do studies like the one you're seeing here. There are a couple of interesting stories that you can tease out of the history of the colonization of the world by human beings just from the genes. And one of them is the neat is we can tell pretty much the route that people took from Africa to the rest of the world. Very interestingly, one of the early splits in the human family tree when you look at any particular locus is between the Aboriginal people in Australia and the rest of the people in the world except for Africans. And here's what seems to have happened. There was a migration out of Africa that then followed the coasts. So people who moved out of Africa into the Middle East ended up continuing down to the east along the coasts. And so one of the first splits was here through the Malay Peninsula down into Australia and surrounding islands. This split happened before anatomically modern human beings even reached Asia. And so this split here is surprisingly old. It's one of the things we learned when we first started looking at these genetic data. Later, there was a second colonization out from Southeast Asia, out to Oceania, heading mostly along the north here. Here's Papua New Guinea and out to the Polynesian islands out here. Another migration from those Asian peoples very recently crossed the Bering Ice Bridge into the Americas somewhere between 15 and 30,000 years ago. And we can get really pretty good dates on a lot of these events just based on genetic diversity among different groups of modern people. One of the most interesting stories we can get from using some of these genetic tools is that there's a surprisingly complicated story about how people got to the Pacific Islands. And these are some of the most remote real estate on the planet Earth. Some of the islands out here in Polynesia are separated by miles and miles and miles of open ocean. And it's remarkable that human beings got there at all. As it is, they got there very, very recently and by some unusual routes. To understand this story, and it's a quick story, but to understand the story, you have to sort of distinguish between two parts of what's called Oceania, which is the whole set of islands that you found all through the southern Pacific Ocean. Near Oceania consists of, well, the islands that are near to Asia. So New Guinea, the Bismarck Archipelago, and the Solomon Islands. These islands here are known as Melanesia. Remote Oceania are the really remote bits of real estate. Way out here, you find Fiji, Samoa, Polynesia, the Santa Cruz Islands, and so forth. But those are much farther out. And they were settled much later. Anatomically modern human beings had reached New Oceania by 30,000 years ago. But remote Oceania, some of those islands weren't settled until 3,500 years ago. So of course, once they had the technology, people sequenced the mitochondrial DNA of islanders living on all these little islands throughout the South Pacific, and they found that those islands had been settled by individuals coming from Southeast Asia. You can see here this red color is the frequency of one particular deletion mutation that's found in the mitochondrial genome only of people who have Southeast Asian ancestry. You'll notice you don't find it at all in Australia. And clearly from the frequency of that allele, these mitochondria here are descended from mitochondria that came from Southeast Asia. What's interesting is that when you look at nuclear markers, the markers that are inherited from both mothers and fathers, you see a totally different pattern. According to these data, only about 30% of the alleles that you find out here in remote Oceania can be traced back to the Southeast Asian mainland. Most of the alleles you see out here come from Melanesia, from near Oceania. and they share a lot more in common with the individuals that you find in Australia. How is it possible that these two genes could basically contradict one another? Well, if you remember, mitochondrial genes are only passed down from mother to daughter. They're only passed on by women, whereas nuclear genes can be passed on by men. And so the hypothesis that geneticists have come up with is that you've got very different patterns of migration by women and by men. You may remember the story about the Belding's ground squirrel, where the females sit tight close to home and the males migrate. In many human societies, you have similar cultural traditions where one sex is expected to leave home and join the family and live with the kin of the opposite sex. When you have that sort of difference in who leaves and who stays, when people get together to form new families, you get different patterns of gene flow, depending on whether you're looking at a gene that's inherited only maternally, like a mitochondrial allele, or is inherited maternally and paternally, as with nuclear genes. So not only are genetics of the present day able to tell us a lot of things about where our ancestors came from and how they got there, they're even able to suggest something about the cultures that those individuals lived in. I couldn't help but add one last use of genetics to settle the question of what people groups came from where. You don't have to look at human genetics to answer some of the questions about how individuals spread across the Pacific Ocean. Instead, you can look at the animals they carried with them. As they settled the islands of the Pacific, modern humans brought with all sorts of other species. Pigs, dogs, rats came along on the ships. There were chickens. And what researchers have done is, in addition to looking at the genetics of human beings, they've looked at the genetics of all of these organisms found on islands across the Pacific. And what they found is a very complicated story. And I won't go into it here, but suffice it to say that the settlement of the Pacific has drawn from both the population that was already present in near Oceania, as well as the more recent settlements by people in Southeast Asia. So again, there are a tremendous number of mysteries. If you are clever and if you understand how phylogenies and molecular clocks work, there are a tremendous number of questions that you can answer about the deep, deep past of our species.